Hello everyone, my name is Nourin Aman. I am PhD candidate at University of the Punjab. Currently, I am working at my PhD project studies on Macromycetes of Pakistan in Dr. Moncalvo's lab at Royal Ontario Museum and University of Toronto. As we know that fungal diversity in many regions of the world has been poorly explored and the geographic distribution of most species or species group is unclear. Now our study tests the hypothesis for one of the lesser explored region, Pakistan, that the climate, ecological niche and lifestyle rather than geographical proximity is a major driver to organismal distribution on Earth. Before going into the detail of the project, let's have a look at Pakistan as an ecologically diverse region. This is the physical map of Pakistan and we can see that it stretches from the coast of Arabian Sea near the delta of River Indus so far north to the mountain ranges of Himalayas and Karakoram. It covers land area of 0.8 million kilometers square, which is 0.67% area of the world. About 58% of the total land of Pakistan consists of mountains and plateaus, whereas 42% are plains and deserts. If we look into biodiversity, the flora and fauna of the country are composed of blend of Palearctic and Indo-Malayan elements, with some groups also containing forms from the Ethiopian regions. I also want to mention here that monsoon season is of immense importance to the biodiversity of the country, especially for macrofungi when mushrooms appear and mycologists can collect and study mushrooms. Monsoon sweeps into Pakistan from the Indian Ocean during early July to the late September. Pakistan covers a number of world ecoregions ranging from mangrove forests stretching from the Arabian Sea to the towering mountains of Western Himalayas, Hindu Kush and Karakoram. Overall, 14 ecoregions are present in Pakistan. The forest types of Pakistan consists of tropical forests with prominent plants like acacia, salix, tilburgia and ziziphus, montane subtropical forest with olea, acacia, barbaris and more. Cedrus, pinus, picea, abies and quercus species are present in montane temperate forests. Likewise, subalpine forests, alpine scrubs and pastures and cold deserts have diverse plant taxa. So in spite of being ecologically diverse, the knowledge of fungi is scarce and scattered, which needs to be compiled. So the aims and objectives of the present project was to expand and improve the information base on the Macromycetes biodiversity and develop a compendium of Macromycetes reported to date from Pakistan, to explore and identify some unexplored taxa from Pakistan, to categorize reported fungi according to their respective ecoregions and provide a conservation baseline for fungi of Pakistan, to compare geographical relationship of Macromycetes of Pakistan with rest of the world. As the first part of our project, we compiled a compendium enlisting Macromycetes, which are fungi visible to naked eye from Pakistan. We collected data from checklists, literature and the NCBI Genbank database. We followed classification system as per Indus Fungorum. We have compiled a list of 1976 species belonging to 276 genera, 78 families and 15 orders so far. A similar study conducted recently for compiling a checklist for macrofungi of South Africa and listed 1008 species. For comparison with a well-studied area, I want to mention here that 3,173 species have been reported from Quebec. Overall, approximately 20,000 species of macrofungi are known worldwide. This is an overview of our compendium. I have not added species name here, but of course we have recorded each taxa in our checklist. As a second part of project, we did sampling of Macromycetes from 4 different sites of Punjab, including Pabbi Reserve Forest from district Gujarat, Shadra and Jhok Reserve Forest from district Lahore, and Dalana from district Dera Ghazi Khan. We published first scientific study on truffles from Pakistan as a part of this project. Truffles are found mostly in sparse irrigated areas of Pakistan, and surprisingly, these are found forming mysterious relationship with sorghum and millet crops. We collected truffles from agricultural crops of sorghum in arid region as well as near acacia in semi-arid region. We proposed new genus of truffles Ahmadiyya in the family Pisciaceae with a single species Ahmadiyya dalanensis. Our studies were based on morphological basis, anatomical features, as well as phylogenetic analysis based on LSU region. We observed the clear and strong position of Ahmadiyya dalanensis here between Stauphera and Dilastria species. We are also working on description of few more new species which we collected. To see the biogeographical distribution of saprobic and ectomycorrhizal macrofungi globally as well as ecoregion distribution within Pakistan, we analyzed few taxa. For this analysis, we pulled out all the sequences of Macromycetes of Pakistan from the Genbank, and for each taxa, similar sequences were retrieved from BLAST in the Genbank, and finally we built the trees based on ITS, which is fungal barcode. So first come Bovista, which is saprobic. 9 Bovista species have been reported from Pakistan. Out of these 9, only 2 have sequence data submitted in Genbank. Genus Bovista have been collected from following ecoregions of Pakistan: Western Himalayan broadleaf forest, Northwestern thorn scrub forest, Himalayan subtropical pine forest, Western Himalayan subalpine conifer forest, Northwestern Himalayan alpine scrub and meadows, and Balochistan xeric woodlands. To compare global distribution of genus Bovista, we can see Bovista plumbea from Pakistan forming a clade with Asia, North America and European taxa, with a sister clade in South America and the second sister clade showing distribution in North America and Europe. Similarly, we can see Bovista himalaica from Pakistan forming Asia only clade with a sister clade showing distribution in Asia, North America and Europe. Then comes Gymnopus, which is also saprobic. 11 species of Gymnopus have been reported from Pakistan, and out of these 11, 6 species have sequences found in Genbank along with 9 Gymnopus sp sequences. Gymnopus have been collected from 3 ecoregions in Pakistan, which are Western Himalayan broadleaf forest, Western Himalayan subalpine conifer forest, and Northwestern thorn scrub forest. Looking at its biogeographical study, we see Gymnopus barbipus forming clade with European taxa, with a sister clade in Mexico and North America. Also, Gymnopus sp from Pakistan is forming Pakistan only clade, with a sister clade from Asia and Europe, and the other sister clade showing European distribution. Now comes genus Amanita, which is an ectomycorrhizal genus. 22 Amanita species have been reported from Pakistan, and out of these, 11 species have sequences submitted in Genbank. This genus is found in 4 different ecoregions in Pakistan, with the highest distribution recorded in Western Himalayan broadleaf forest, and lowest distribution is recorded from Northwestern Himalayan alpine scrub and meadows. To see the biogeographic comparison of genus Amanita, we can see Amanita ahmadiyyi from Pakistan is forming a Pakistan only clade with a sister clade in Asia only. However, Amanita orsonii from Pakistan is forming an Asian clade with a sister clade occurrence in North America. Genus Suillus is ectomycorrhizal, and a total of 15 Suillus species have been reported from Pakistan. Out of these, 5 taxa have sequences records in Genbank. Genus Suillus have been collected from 5 different ecoregions in Pakistan, with the highest occurrence recorded in Western Himalayan broadleaf forests. The biogeographical analysis of genus Suillus shows that Suillus americanus from Pakistan and Suillus sibricus from Pakistan nest in a huge clade with Asian and European occurrence, with one sister clade showing North American distribution and the second sister clade showing Pakistan distribution. On the other hand, Suillus marjani elevatus from Pakistan is forming an Asian clade with a sister clade 1 from Pakistan and the sister clade 2 showing Asian occurrence. So overall, when we put together our results, we saw that in case of saprobes, major clade has North American occurrence just once, while 50% of times we have presence of European taxa. On the other hand, ECM clade is 50% Pakistan only and has 20% European representation. Additionally, the sister clade has 70% North American representation in saprobes and less than 50% in case of ECM genera. So our preliminary results of biogeographic relationships using the ITS DNA barcode marker indicate that ectomycorrhizal taxa are more restricted geographically than saprophytic taxa. ECM are significantly more abundant in the northern regions and show pan-Himalayan and European relationships, which are temperate climate range, whereas many species group of sap tend to be more widely distributed globally. Thanks for watching, and if you have any question regarding my project, you can contact me on my email address.